Fishery Science Center. This is the third of 9 weekly groundfish seminars running through December 13th. Since we are all doing this seminar remotely, the speaker will use the electronic laser pointer or be descriptive when indicating specific points on the slides. To help with this format and to avoid additional distractions, please mute your audio and turn off your video feed. Also, please keep your questions for the end of the seminar, or you can type them into the chat box if you think you might forget them. And at the end of the seminar, Bianca and Sarah will be fielding the questions. Before I introduce today's speaker, I want to remind people to join us for next week's speaker, Kathleen Vestfals from the Fishery Resource Analysis and Monitoring Division at the Northwest Fishery Science Center, who will talk about identifying stage-specific drivers of Pacific hake recruitment, which will be held at the same time next Tuesday, October 25th. Today's speaker is Thaddeus Buser. Thaddeus received his bachelor's degree in aquatic and fisheries science from the University of Washington, his master's degree in fisheries from the University of Alaska Fairbanks, and his PhD in fisheries from Oregon State University. His research interests center around the biology, ecology, and evolution of North Pacific fishes and invertebrates, with a particular enthusiasm for sculpins and their relatives. Before joining the Alaska Fishery Science Center, Thaddeus was a postdoc research associate at Rice University, where he studied the diversity and evolution of fish skulls. And today, Thaddeus will be talking about the morphology and evolution of cranial weapons in sculpins.
Okay, thanks, Mark. So playing before you is a video of 2 sculpins. Specifically, these are snub-nosed sculpins, Orthinopius triasis. And what you're seeing is 2 males, and they're coming up to each other, and they're kind of saddling up to each other, nipping at each other's faces, and then shaking each other around kind of cheek to cheek. And how they are doing this, and especially why they are doing this, is going to be what I'm going to talk about today. Before we get into that, though, let's talk about something that might be a little more familiar to people. This is maybe not this individual species, but the deer family, Cervidae. What you're looking at here is a muntjac. They are about the size of a medium-sized dog when they're full grown. And uniquely among living members of the deer family, they have a peculiar kind of weapon that comes off of their face, and that is these gigantic elongated fangs. And they have a unique fighting style to go along with this. Male muntjacs during the rut will engage in this fighting where they're basically trying to bite each other in the neck with these big fangs. And it turns out that they have all kinds of, strategies for doing this, and they even have evolved these bony placodes in their neck to basically protect their most vital parts from getting chomped on by another individual during these fights. But it turns out that they have a pretty adept sense of when the other male is going to try to bite them, and they're very, very fast, and they're good at avoiding getting bitten. And so they have this unique kind of goofy looking antler set up here that you might have noticed. And what they use these antlers for is actually for trying to kind of knock the other male off of their balance, so that they can, while they're kind of stumbling and trying to get back, standing again, they can lunge in and go for this big bite to the neck. And this is not something that you see in other species of living deer, but it's thought to be kind of how deer evolved antlers and how they're fighting, and kind of the basis of their fighting style that's then evolved into much more of kind of a grappling, combat where they're locking antlers and using, basically it becomes a test of strength rather than a test of who can bite each other in the neck first. And this kind of, head-to-head fighting over a limited resource is basically how we think that most weapons that project off of the head of an animal came to be. And this is especially true in terrestrial environments. So everything from the awesome antlers of Megaloceros, the Irish elk, to dinosaurs, Styracosaurus is my personal favorite, to living bovids, as exemplified here in these rams, and even things like elephant tusks. All of these big weapons that project off of the head of an animal are thought to have evolved primarily as a tool in intraspecific combat, so fighting other individuals of the same species for access to some limited resource. And because this has happened over and over and over again in all kinds of independent lineages, there's been a couple of trends that have kind of come out. One of them is that weapons tend to evolve primarily to as part of these intraspecific competitions. So males fighting over females, females fighting over males, males fighting over territory that they then use to lure in females, or females fighting over territory that they use for getting nutrients during the wintertime in the case of like caribou. There's also this ontogenetic shape change where you have young, immature individuals with relatively small, relatively simple weapons or no weapons at all. And then by the time that they grow up into becoming reproductively mature, they have their kind of full set, the most elaborate form that the weapons will take. And then you also have these really high rates of asymmetry where the one side of the head can have a very different shaped weapon than the other side of the head. And in some species of deer, for example, this is basically fixed, where they have kind of this plate that grows to protect their eyes. But often it is a case of kind of asymmetry that doesn't really seem to serve a purpose. It's thought to be reflective of kind of a genetic bottleneck on really, really high, really strong sexual selection on these traits that then kind of leads to kind of a shallow gene pool for growing these weapons. And this isn't just a vertebrate thing. This is widespread throughout invertebrates as well. This is actually a wasp, the properly named tusked wasp. You find weapons evolving all over the place in terrestrial animals. You also find weapons evolving all over the place in fishes. And in fishes, though, they don't tend to project off of the head the way that they do in terrestrial animals. You see more often modifications of like the fins, in the case of catfishes and rockfishes, or in scales or scale support systems like you see in rays and skates, and like in porcupine fishes, things like that. And what's interesting about this is that even though we have, this incredible diversity of weapons that have evolved in fishes, very few people have really paid much mind to why these weapons evolved in the first place. The obvious kind of default answer is that these things serve to protect them from predators, and certainly they do that, but whether that's the only thing that they're used for or whether that's even the primary thing that they're used for has not really been tested in more than a handful of species. Now, of course, I'm going to be talking about sculpins, a group that I'm especially partial to. And sculpins have, as anyone who's ever come across them knows, these big projecting spines that come off of their faces. This is a modification of the preopercle bone. If we peel back the soft tissue here and look at the bones underneath it, this is a CT scan, a reconstruction from a CT scan of the Leicester sculpin, Anopheles lucasi, a North Pacific denizen. You can see that the preopercle bone here, which is kind of rotated forward, has multiple spines that are coming off of it. And there's even spines that are coming off of the spines from the preopercle. And not only is the preopercle weaponized, but all kinds of other bones have been weaponized as well. Everything from the lacrimal to the to elements of the neurocranium, even the suborbital stave, which is kind of a weird bone to begin with, has been turned into a weapon. And so sculpins have really clearly put a lot of energy into developing these spines, maintaining these spines, and so they must be using them for something important because these are not cheap to grow. So to get at the first part, the first question of my talk today, how are sculpins actually deploying these spines. And then later on, we'll talk about why sculpins are deploying these spines. So for the how, though, like clearly these things can point out in this kind of very projected way from the rest of the body. This is not something that most fishes can do with the preopercle. The preopercle is part of the opercular series, so it's part of the bones that fishes use for breathing and for sucking in prey and for kind of moving the mouth to a certain extent. So these are not bones that don't serve a purpose. There's something important that's going on here outside of these spines, and so how would sculpins kind of get around that to use them as these weapons? Sculpins have an extremely enlarged muscular system that actually attaches to the preopercle. So in purple here is the levator arcus palatini. In most fishes, it's this really minor kind of ribbon of muscle, but in sculpins, it's this massive, massive sheet of muscle. There's also the adductor mandibulae that attaches to the preopercle, and the adductor mandibulae is typically thought of as something that moves the mouth primarily. And attaches to the preopercle basically as support. So I went in and wanted to test whether these muscles actually can move the preopercle forward in kind of that threat position that we typically see them in when we're handling them. And so I got a big old great sculpin, Myoxocephalus polyacanthocephalus, euthanized it, and then attached some electrodes to it. So we're getting close to Halloween, so I thought this kind of Frankensculpin would be a perfect thing to talk about today. And this is me stimulating the levator arcus palatini with electricity, and you can see clearly that on this dead fish that the preopercular spines get projected out. And we tested a whole bunch of other different muscles that are associated with the head, and nothing else would project the preopercle out like this. And so we kind of came to the conclusion that yes, in fact, this gigantic levator arcus palatini that's only found in sculpins in this size is in fact the thing that they're using to project it forward. But I wanted to dig a little bit deeper into kind of the mechanics of how this all works. And so I adapted some technology that you're probably familiar with already. Lord of the Rings came out a long time ago at this point, but this is also the same technology that they use for any kind of motion capture in Marvel movies or any other kind of computer assisted animation. So basically, you know, in Lord of the Rings, they dress up an actor, Andy Serkins, in this kind of lycra, super, super clingy suit and attach all of these white kind of balls to it, to the outside. And so then as he's moving and kind of going through playing the part, they film it with two cameras that are at 90-degree angle to one another. And then what they can do is they can track the movement of these balls in three-dimensional space using this two-camera setup, and then kind of reconstruct the movement of each of those balls in 3D space, align that to an animation where you say, okay, this ball that's, you know, on his bicep here is the same thing as this point on the animated figure. And then you can have the animated figure duplicate exactly the motion of the actual actor with really high precision. And that's how they made Gollum, and that's how they do the Incredible Hulk and things like that. I used that same technology, but I did it with a fish, and I didn't use a lycra suit to attach the balls. I did something a little more invasive. I actually surgically attached the balls. So here is a CT scan of this buffalo sculpin that I looked at, and actually surgically implanted these white balls that show up not only in video but also in X-ray. This is, you know, a reconstructed series of X-rays basically, and then that allowed me to basically do exactly the same thing that they do in Lord of the Rings. I prompted a sculpin, to this buffalo sculpin, to go into its kind of preopercular flaring movement, and then used that to animate a model of the skeleton that I built from a CT scan of the same fish. So an extremely precise model of the skeleton coupled with this extremely precise movement of these points in 3 dimensions to recreate what those bones are doing when the sculpin goes into these flaring motions. And so this is this this 3D animation of the sculpin going through this flaring motion. And if I can get it to fire one more time here. These bones will rotate forward and kind of independently of the rest of the skull, this preopercle is actually moving, rotating out away from the plane of the body. And it's not a movement that other fishes are capable of. And sorry, I only did it one time. I don't know why it's not replaying it again, but alas. Another way that I looked at this same problem is actually whether I could get the preopercle to do this movement away from the skull using just the arrangement of the bones of the skull. So this is also a recently euthanized great sculpin. I've removed the skin, and I'm just holding the mouth shut with a pair of forceps here and pulling back on the pelvic fins, which kind of pulls the whole lower part of the jaw opening mechanism, this hyoid arch here, pulls that down. And basically what I discovered is that these sculpins, in addition to using this levator arcus palatini muscle to rotate the preopercle out, can also rotate it out simply by clenching their jaw and going through the same kind of motion that they would use to otherwise kind of open their jaw. Fishes have kind of two major sets of bones here, and if they keep one of them shut but open the other, it actually projects out that preopercle as well. And actually, when they do that, it puts the preopercle in kind of this very rigid attachment where it, or this rigid position where it's being essentially buttressed by this suborbital stay, which is a bone that you don't find in a lot of fishes, but you do find in sculpins, and basically creates this kind of diamond-shaped structure that holds that preopercle out in its projected position. And so this is something that they can accomplish in multiple ways, and then once it's projected out, it is held out really, really robustly. And this is important, obviously, if you're using it defensively. So here is Myoxocephalus again, this time in the jaws of a red Irish lord. And if you look closely, you can see that the preopercular spines are sticking out and kind of pinning into the jaws of the Irish lord here. What the ultimate fate of this particular great sculpin was in this scenario, I can't attest to, but I can attest to the fact that we do find sculpins that are with their preopercular spines sticking out in the jaws of dead big fishes, like, for example, a lingcod. Lingcods are a major predator of sculpins, and we actually have a CT scan of a dead lingcod that washed up that had a great sculpin partially down its throat with the preopercular spines sticking out into its gills. And so they clearly are an effective means of getting of not being swallowed, or at least taking down the predator with them. So all of this kind of supports the notion that like it's presumed in a lot of other fishes, that these preopercular spines have a major defensive component to them. But that's not the whole story. And what we'll talk about now is why do sculpins deploy these spines? We've just talked about kind of how they do it, and some evidence that this is a really strong defensive structure that clearly can be used defensively, but there's been a lot of observations that make the story more interesting. So this is an illustration, it's one of my favorites, from Morris, 1956. And this is a sculpin. This is the European bullhead, Caudus gobio, and it's a male, and it's sitting in this little cave. And they do this during the breeding season. They will find these little caves, kind of clean them out a little bit, and then sit with their face poking out of it, waiting for females to go by in the hopes that a female will want to come in and lay her eggs in this cave. And often, though, other males will come by, and when they do that, the sculpins will engage in this threat display, and which Morris termed as the threat attitude of Caudus gobio, with the preopercular spines flared out and this kind of characteristic, big, bad looking face behavior that they engage in. And so even though there is clearly no predator around. This isn't something that they're doing to avoid predation. This is clearly something that they're doing to intimidate other individuals of the same species, intraspecific combat. And that kind of puts them, in contrast a little bit between what we typically think of as, fish armor and, why they evolve weapons, these spines that are against predators versus how terrestrial animals tend to evolve weapons, which is to settle intraspecific disputes. And so that sets up kind of this, stag hypothesis of intraspecific fighting versus the pinecone fish hypothesis of defending themselves from predators. And if you recall from the beginning of this slide or this talk, we had this video of these two snub-nosed sculpin coming up to each other, biting each other in the face, and then kind of saddling up cheek to cheek and shaking each other around. And this is a CT scan of what the preopercular spine actually looks like in a snub-nosed sculpin. And you can see that it has these kind of big, long branches and all of these kind of recurved hooks. And this is exactly what you would expect if they were using them to try to tangle with one another the way that deers do, to be able to shake each other around as they appear to be doing in this video. There's more evidence that kind of suggests that sculpins are doing more with these spines than just defending themselves. This is a male scaly-head sculpin, Artedius harringtoni, another local fish. And I found this sculpin actually has kind of a sad story to it. So I had set up this tank of sculpins in this laboratory. I was working in my lab late one night, and each fish in the tank had a rock that it was kind of its little territory, and it would sit on the rock, and if another male of the same species came around, it would chase it off. And everything was kind of at peace because every male had a rock that it could sit at and defend. And during the night, it seems somebody was setting up another tank and took away one of the rocks from my tank so that they could use it in theirs. And so we had this now mismatch between the number of males and the number of rocks. And when I came in the morning, this poor individual was dead. And being the curious biologist that I am, of course, I dissected it and found that it had all of these puncture wounds around its face, especially around the cheek. You can see kind of the red here from all of the different punctures on its face, kind of where you would expect punctures to be if it was engaging in a behavior like those snub-nosed sculpins. And it turns out that scaly-head sculpins actually have a lot of sexual dimorphism in the species. This is a male here on the left and a female on the right. And you can see right off the bat that males kind of grow these big, fluffy kind of plume-looking cirri above their eyeballs. They also have these enlarged mouths with these giant canine teeth that are not something you find in females or juveniles. And then during the breeding season, males actually develop these spots on their anal fin that happen to look an awful lot like the eggs of scaly-head sculpins. And this is a behavior that has evolved multiple times in fishes where males that have nests, it looks good if they have lots of eggs already in their nest. And so they will tend to develop these anal fins with spots on them that look like eggs. And when a female comes around, the male will kind of splay that anal fin out. So it looks like there's all of these eggs in its nest. And females will see that as kind of oh, this must be a good place to lay eggs. So a bit of subterfuge there. So that got me thinking about, given that there's this evidence of combat, this evidence of at least kind of antagonism between males, there's evidence of sexual dimorphism, would we, if we looked at the preopercular spines and the shape of those spines, would we see the same kind of patterns that we saw or that we see in terrestrial weapons, like we see in deers and things? So would we see asymmetry, high rates of asymmetry? Would we see sexual dimorphism in the shape of the weapons themselves? And would we see ontogenetic shape change? And if we see all of these things, then that would be really good evidence that actually these sculpins might be the stags of the sea, and they might be using their preopercular spines, at least in addition to defense, in these offensive kind of contests. So what I did is I CT scanned a whole bunch of fishes so that I could make really highly precise digital models of their skeleton. Most species of sculpins don't get very big. The head of this fish in particular that I have here, the total length of this fish was about, 3 inches long, 2.5 inches long. So the head's about 1/2 inch long, and the preopercles, like 1/4 inch long. These are quite small. But if you CT scan them and you build these precise digital models, you can scale them infinitely, and so you can make it as big as you want it. So that's what I did, and then I kind of found these places along the bone that are consistent across all of the species, and compared those places quantitatively so that I could kind of describe the shape and compare it in a quantitative sense across species. And so it turns out the sculpin preopercles are hugely diverse in terms of their morphology. Often the shape can be specific to a genus or sometimes even a species. If you're familiar with some like fishes of Alaska and using out, if you've ever had the pleasure of keying out some sculpins using that, preopercular spines can often be one of the most important diagnostic features. If you look at spine shape across all sculpins, it varies from these one of the biggest ways that it varies is kind of the length and straightness of the uppermost spine, and associated with that are kind of this L-shaped preopercular bone. So you have this kind of short part of the L at the top and the long part of the L at the bottom. On the other end of the spectrum are these kind of short uppermost preopercular spines, and in those you have this much more crescent shaped shape of the preopercle associated with that. And this accounts for about 40% of all variation in shape across 150 species of sculpins that I looked at. And if we look at kind of a shape space, so here is that, long pointy spines versus short spines on this long axis here, this horizontal axis. And then on this other axis here, I have a different aspect of shape. This is basically the curvature of the uppermost spine along with the length of the second spine. This is kind of how these species fall out. So things like the genus Anopheres, the buffalo sculpin, and its relatives have, as everyone who has seen them knows, these very long spines. As you get into things like the freshwater sculpins, the spines are much shorter, and there's a lot of variation within that. If we want to zoom in here on one of my favorite groups, this is the intertidal sculpins. We'll look at them as kind of a case in point for broader trends here. I'll talk about a data set here that consists of the 17 species in the subfamily Oligocotini, which includes all of the intertidal sculpins that occur around here. I looked at 8 to 32 individuals per species, and I looked at both sides of the head, and for each fish I actually did 3 replicates. So I made a digital model of one individual and then made 3 copies of it, and then looked at the left and right hand side of each of those copies. So altogether I looked at around 1,400 digital models of these preopercular spines and the underlying preopercular bone to kind of tease apart some of the story here. So we predicted that, if sculpins are using their spines offensively, that there would be substantial bilateral asymmetry like you see in terrestrial animals. And it turns out that that is the case. Here's 4 individuals with their left and right preopercles. So the way that they vary in shape kind of varied between how long the spines were on the uppermost. So this one has multiple spines that kind of project from the upper spine. And you can see that on the left-hand side of the fish here, its left-hand side, the spines are long and pointy. On its right-hand side, they're kind of reduced and nubby. We also had variation in the number of spines. So on this individual here, there's 1, 2, 3, 4 spines projecting off of that upper spine. On its left-hand side, on its right-hand side, there's only 1, 2, 3. You also had variation in kind of the direction that the spines are going. So on this individual here on the right, on its right-hand side, the spines are pointing upward. On its left-hand side, the spines are pointing downward or backward. And you also had some variation in kind of more complex aspects of the shape of these spines. So the left-hand side here has longer, kind of wider spines than the right-hand side. And if we look at some of the kind of fine scale variation here, so this is kind of, looking at it at a big scale, and you can kind of appreciate there's differences in shape. But if we take full advantage of the highly precise nature of these digital models and the methods that I was using for kind of quantifying shape, you can get it a little finer. So what I have here is I have each point represents a digital model of a preopercle. And for a given individual, the preopercle that's on the left, the three replicates of it are represented in green, and the three replicates of the right preopercle are represented in purple. And then I have an arrow that points from the left side to the right side for a single individual. So this is a single individual, this is a single individual, this is a single individual. And you can see here that my replicates group together very tightly and much tighter than the left-hand side versus the right-hand side. And so this tells me that this difference that I'm seeing between the left and the right is not just an artifact of how precisely I placed these points or how precise my digital model was. It's actually a much greater difference than the replications I did. And I tested this, statistically, and it's all significant. So this kind of gives us an ability to look at asymmetry in a more quantitative sense. So for example, here's an individual, and you can see the left-hand side here and the right-hand side here. And here's another individual, and the left-hand side is here and the right-hand side is here. And what you can appreciate from this is that the right-hand side of each of these two individuals is much more similar in shape to the right-hand side of the other individual than it is to the left-hand side of that same fish. So the right-hand side of a fish's face looks more like the right-hand side of some other fish's face than it does the left-hand side of that same fish. And if we look at this across all of the species, you can actually find individuals here where the left-hand side of a fish is more similar in shape to the left-hand side of a fish from some other species than it is to the right-hand side of that same fish. And so it turns out that asymmetry is really, really drastic in the preopercles of sculpins. And much more so than I was really anticipating when I kind of set out to see whether this was the case. And not only is it bigger, not only is it an issue within a given species, but it's actually an issue across species where you can have shapes that don't look like the same species on either side of the same individual fish. And this is a problem for people like me who are interested in kind of the evolution of traits, but it's also a problem if you want to think about, what the shape of a fish, how big its mouth is, how big a preopercle is, how big the opercular apparatus is on one side of a fish. If you want to assume that you can measure traits on one side of a fish and that's representative for the entire species, it might not even be representative for that individual fish. And so, for example, here, I'm kind of doing one of my analyses here, looking at those same two axes of variation, that kind of spine elongation versus kind of spine curvature here, looking at the same set of species, and we'll just map out a few individuals. So here's one species, Artedius fenestralis, another species, Oligocatus rimensis, and a third species, Clinocatus acuticeps. This is using only left preopercles to kind of make this look at this plot. This is only using right preopercles, and you can see that the distribution of these species is quite different in these two plots. And so this is concerning because depending on whether you're looking at the left-hand side or the right-hand side, you can get different answers to the same question. And this is actually using the same individual fish. So this isn't just like me trying to find the most extremely different within a species. This is looking at a single fish, and here's where its left side falls out in this plot, and here's where its right side falls out in this plot. And that's the same for all of these. So you can get very different answers depending on which side of the fish you're looking at. So the solution to that is to look at lots of fish and to take averages if that's appropriate. Sexual dimorphism, it turns out, surprising to me, was really not hardly any evidence for it. This is a single species that I'm using kind of as an exemplar. This is that snub-nosed sculpin that we saw the video of. I have females here in blue, males in orange, and you can see that the shapes of the preopercles are variable in either sex, and there is no kind of bias in shape given a sex. And that was the case for all of the species that I looked at. I did find really strong evidence of ontogenetic shape change, though. So small individuals having, in this case, more simple but preopercles, and as they get larger, developing these more complex and relatively larger spines on their preopercle. And this matched kind of the expectation for species that are using spines for intraspecific competition, because sexually mature individuals would have a good reason to want the biggest, most complex spines possible, because they're the ones that are using the spines for combat. But I also found lots of evidence of the opposite, where spines actually started off in very small individuals. This one is just barely post larval juvenile, and the spine is relatively elongate. This is looking kind of like the buffalo sculpin spines. And as the fish gets bigger, the spine actually gets reduced in complexity and relative length to becoming just kind of this nub that pokes out. And even the shape of the underlying preopercle bone changes in that same kind of way that we saw it, across species. It actually changes in the same way across ontogeny in most of these species, where it becomes more crescent shape as the spine becomes smaller and nubbier, having started off in more of an L shaped when the spine was elongate. So, to kind of gather everything together here, our sculpins, the stags of the sea, it's kind of, we're still kind of trying to sort that out. So there's no sexual dimorphism, but we did find really strong evidence of ontogenetic shape change, and we also found really strong evidence of bilateral asymmetry. So whether they're using their spines, to fight within a species, individuals of their same sex, there doesn't seem to be any evidence in the spines themselves that males and females are using them for different purposes. This doesn't necessarily exclude the idea that they could be using them for different purposes. It may simply be that selection hasn't really caught up to what they're doing with them now. It also could be the case that both sexes have things that they're doing with the spines that aren't necessarily the same, and we just aren't appreciating, the way that females might be using them. They may require the same kind of shape that males do, but they might be using them for a different purpose. We simply don't have the kind of long hours of observation of exactly what these fishes are doing underwater with these spines to kind of tease apart that more at this point. So with that, I'll just acknowledge a lot of the individuals and funding sources that have contributed to what I've shown you today. This is kind of a long effort that has gone through multiple institutions and has gotten supported by various funding agencies, and had lots of support from other researchers that I've been fortunate enough to work with. And I think I have plenty of time for questions if anybody would like.
Wow, very cool research. Thank you for sharing that with us, Thaddeus. And I will never look at a sculpin the same way again. And now we're going to hand it over to Bianca and Sarah to field questions from the chat or from people's microphones.
It looks like Sean Rohan has a message in the chat. That's super interesting talk. It seems like some of the mechanisms you've described for competition between males depend on visual cues. Since visual cues are more likely to be available in shallower habitats, would that imply armoring would be less ornate in deep water sculpins?
Yeah, great question. In fact, you do see that in a lot of deep water species, the preopercular spines are really, really reduced, sometimes completely absent. So the blobfish and the blob sculpin are both a kind of sculpin. They're kind of a weird group, a subfamily within sculpins broadly, within the marine sculpin radiation, and their preopercular spines are practically non-existent. And they're generally kind of poorly ossified as a group. Now, whether that is a function of, predominantly losing kind of visual cues that would lead them to not fight each other so much versus kind of more external factors like the difficulty of, kind of maintaining ossification at very deep depths, that's a harder thing to tease apart. Sculpins certainly do get very deep, and most of those deep fish tend to have small preopercles, but there's, like all good trends in biology, there are always interesting exceptions to that rule. Like one of the few sculpins that lives in the southern hemisphere has massive preopercular spines, and it lives in super, super deep depths. Maybe they're using them for something altogether different. I'm not really sure, but that's a great question. And again, kind of settling that would be hard to do without a lot of ROV hours, I suspect.
It looks like Dwayne has his hand raised. Would you like to ask a question, Dwayne? Unmute yourself.
Yeah, thanks, Bianca. And thanks, Thaddeus. That was great talk. It was a little troubling, though, in some ways, because, of course, we use preopercular spines and all sorts of cranial weapons as identification characteristics for sculpins and for a lot of other species as well.
Yeah.
So that's a little problematic. But I'm also wondering, is there any information on, the sorts of asymmetry, sexual dimorphism, ontogenetic change that you noticed in cranial weaponry? Is there any information about that sort of variation in scale development?
Great question.
Because we use scales, the placement and the size and shape of scales
Yeah.
as identification
Yep.
characteristics for a lot of the species as well.
Yeah. Scale patterns can change in some species across ontogeny, but I think that it's pretty well documented. Whether that information has been relayed into, guides and keys, of course, is a different question altogether. But for practical application, the individuals that we're going to be coming across predominantly are going to be sexually mature and are going to have one end of kind of that distribution of potential sizes, shapes, etc. And so what we're looking at is probably not going to be hugely affected by this ontogenetic shape change. Most of the change is happening between kind of the earliest juvenile sizes, post-larval juvenile sizes, and getting up to sexual maturity. And most of these species, sexual maturity happens pretty rapidly. So I don't think we have too much to worry about in terms of what we're doing. But certainly people who are into, like CT scanning and other things where you're size limited in the individuals that you can look at, they really need to worry about this kind of thing.
Do we have any other questions? I don't see anything in the chat or any hands raised, but feel free to raise your hand and unmute yourself.
Did anybody think that, before this seminar, did anybody think that Andy Serkins could be related to Sculpin functional morphology? There's always that 6 degrees of separation, you know.
I'll ask a question. Are you going to apply these same methodologies to other taxa, or where's the future for this work?
Yeah, so the great part about everything that I presented here is that it's based off of kind of these digital models of the skeleton, from CT scanning. And the cool part about that is that these digital models of the skeleton are basically, as long as you back them up somewhere, they're indestructible and they're infinitely shareable. And so everything that I presented here is actually available for free up on this website called Morphosource. And that has been kind of a push among a lot of anatomists recently, is to kind of CT scan species and put them up on the internet for free. And there's a movement called the Scan All Vertebrates movement, and the idea there is one of the components of it is scanning all the fishes, and that is the push there is to scan individuals of every family, if not every genus, and get them up on the internet. So I've CT scanned every species of sculpin that is available in North America in a museum somewhere, and even some that are from Europe and that are from European and Asian museums, and put them up on the internet. And that's also the case for a lot of North Pacific fishes that other researchers have done. And so these methods can be applied to any kind of piece of anatomy, and they're really great for looking at CT stuff. And CT stuff is freely available for a lot of North Pacific fishes already without any extra additional work.
We have a question from Lewis in the chat. He said, surely, I guess there's two. Interesting how the armoring story relates to something like the stickleback. Can we infer how fast armoring traits evolve between these two? And then surely the stickleback evolved faster, but it's proportional to their generation time, or is it disproportionately greater due to the strength of selection pressure?
Yeah, great question. So the great thing about sticklebacks is that we have all of this temporal information to go along with the morphology. And that's not so much the case for sculpins. Even the fossil record for sculpins is very poor once you go back past, the Pliocene. It basically goes away. All the fossils that we have are mostly from extant genera, and there isn't anything at the scale that we have for sticklebacks. Certainly no one has watched them, lose or gain armor the way that they have sticklebacks, over the course of a human lifespan. Sticklebacks, I think that they're under a greater kind of selective pressure because the change is so drastic when they, move out of the ocean into freshwater. If anyone isn't familiar with the kind of pattern there, sticklebacks will invade freshwater all the time. They'll move, there's a huge population of them in the oceans, but they're constantly invading freshwater, and sometimes they get cut off in some freshwater system like a lake. And when they do that, they're all of a sudden in this environment that is not only limited in calcium ions, but also does not have anywhere near the selective pressure typically that they're facing in the ocean. And so they tend to lose their armor very, very rapidly. An ocean stickleback will have these massive plates of armor on their sides and these big, robust fin spines. hence their name, 3-spine stickleback, 9-spine stickleback, 12-spine stickleback, all the spine stickleback. And they lose that really rapidly in freshwater. And was this interesting kind of case in Lake Washington where they're under predation pressure from cutthroat trout. And historically, the city of Seattle and Bellevue would just dump their raw sewage into Lake Washington, and it became eutrophied. All of the visuals completely went away. There was no visibility. And all of a sudden, these stickleback lost all the predation pressure. They lost all their spines. They lost all their armor. And then we cleaned up our act. The water got clear again. And then they re-evolved all of their spines and armor. In the course of like 50 years, that whole cycle happened. And so they're clearly able to do it very, very rapidly. But again, I think that the selective pressure that they're under is much stronger than, like the incremental pressure that, a sculpin fighting another sculpin, if it grows an extra spine on its preopercle, that's going to give it a very slight advantage compared to like rearmoring itself to avoid being eaten by a cutthroat trout. I think that you're right that the pressure differential is far greater for stickleback.
Looks like we have another question from Dwayne. What's the potential for CT scans as identification tools, particularly with respect to stomach contents and other degraded specimens?
It's outstanding. So preopercles are one of the ways that we identify sculpins, this fluctuating asymmetry notwithstanding. And preopercles, turns out, are one of the things that stay longest, in the stomach, because they're heavily ossified. So they take longer to kind of get demineralized and absorbed. They also show up in middens, in archaeological middens a lot. And again, they're kind of are the thing that sticks around for a really long time. And so having a database of preopercles for different species and actually having this kind of quantitative shape space of where different species lie and the kind of, being able to use numbers to characterize the shape is great, not only for comparing them, but it's also great for kind of placing them. And so if you had an individual who came across a sculpin preopercle and had no concept of greater diversity in sculpin preopercle shape, but could put points on a bone, if they knew where to put those points, they could hypothetically do that, enter it into a system, and get a confidence of species ID that would be pretty good. And then, from there, they could probably figure out exactly what it is based on a few other elements, if they knew where it was collected or things like that. So the potential for using this kind of technology and this approach to looking at shape specifically of the preopercle has a lot of applications beyond just, hey, isn't that cool kind of science. There's also a lot of real practical applications for species ID.
Any other questions?
I think you're muted, Mark.
Yeah. So your work was showing a difference between left and right sides of the sculpin skulls. So is this an indication of right-handedness or left-handedness in these fishes that we didn't know about before?
That's a great question. And that's like one thing about it, though, is that there's high levels of asymmetry within a species, but it's not, consistent. It's not consistently that the right-hand side looks like X and the left-hand side looks like Y. It's that they're consistently different. And that was exactly what I was thinking too. As soon as I saw how asymmetric they were, the very next thing I did is say, okay, well, are all the left-hand sides, consistent within a species? And it turns out that they're not. It seems that, they are, an individual is potentially doing something handed, but not everybody is the same handedness in a given species.
Thaddeus, do you know where to get a CT scanner on the cheap?
Yeah, you can CT scan a fish for free. At Friday Harbor Labs, you don't even have to go there. You can send them to Adam Summers. You can also send them to Corey Evans at Rice University. They're both operating under the kind of send us your fish and we'll scan them for you. We just want these scans to be available for everyone. And if you write a paper and want to include us as a co-author, that'd be cool too, but not required. So you can scan them for free as long as you're not trying to do 10,000 of the same species. There are multiple labs that are happy to do it for you.
Very cool.
But if you're interested, if you've got like a pet taxon and you want to know what its skeleton looks like, go to morphosource.org and type in that species, and it might already be up on the internet. And you can even, their web browser now lets you kind of even play around with the like a reconstruction of the scan just from your browser. So you don't even have to have the fancy software. If you are interested in doing something more than that, I actually wrote a workflow that uses only free software and it's cross-platform, so it doesn't matter what kind of computer you have. The only caveat is it can't be a government computer because it's free open source software and they don't like that kind of thing. So.
Well, it looks like we have exhausted the questions, unless you see any more, Bianca.
I do not see any more.
Okay. Well, thank you very much, Thaddeus. Excellent presentation. Just great visuals and just really fun to see the science coming out of the imagery.
Yeah, thanks. My pleasure.
So I think we can end the seminar recording there and hopefully see folks back here next week for seminar number 4.